Hello, everybody. So sorry I couldn't attend the last two meetings. So if you saw my post on Classroom, we're going to do a 2-hour class today, just to make up for the lost time. So I'm just going to start sharing my screen and we'll get right into it. Okay, so we're going to be talking about cells. So I guess you could say the main principle of cells is what we call cell theory. So cell theory just says that first thing is that the cell is the basic unit of life. What that means is, like, if anything is living, it has to contain a cell. So some organisms are unicellular, which means that they're made up of only one cell. And then other organisms are multicellular, which means they're made up of multiple cells. So for example, bacteria are usually unicellular, and most plants and animals are usually multicellular. So 3 main points to cell theory is that the cell is the basic unit of life. All living things are composed of one or more cells, and all cells come from pre-existing cells. So, and we talked about the cell being the basic unit of life. If anything is living, it has to have a cell. And then all cells come from pre-existing cells. So when we get into cell division, we'll talk about how cells are formed. But cells can't just spontaneously generate in like the atmosphere or something. They have to come from a pre-existing cell. So skip that, and we'll go into animal cells. So the cell itself is made up of little organs or organelles. So if you remember how we talked about roots last class, E-L-L-E, is the root in French for little. So organelle just means small organ. And so there are microscopic structures inside the cell that carry out the cell's business, if you will. So there's a ton of organelles, and this is what you need to know. So the outside of the cell, which is shown like around here, like this thin green layer, is what we call the cell membrane. So the cell membrane kind of protects the cell. It's the outer layer of the cell. And if you remember from our discussion on phospholipids, the cell membrane is actually made up of phospholipids. And if you remember, phospholipids are the one with the polar head and the non-polar tail. And so they're kind of amphipathic molecules, meaning they have 2 charges. They're polar on one side, non-polar on the other. So the cell membrane is made up of these amphipathic molecules. So the cell membrane actually holds in what we call the cytoplasm. And the cytoplasm you can just think of as the liquid of the cell. So all organelles and every chemical molecule in the cell kind of just floats around in this watery substance called the cytoplasm. It's kind of like a gooey gel, and it just holds everything in there. So the cell membrane, another function of it is it regulates the materials that can flow in and out of the cell. So certain materials, certain molecules can't get into the cell, and certain molecules can. So we call the cell membrane selectively permeable, which means that some molecules can get into the cell. It's very selective with which can, but a lot of molecules cannot. That's why it's selectively permeable. So the next major organelle is the nucleus. And the nucleus kind of controls all of the cell functions. It's kind of like the brain of the cell. And it also contains the genetic material, the DNA. And if you remember, the DNA is packaged into something called chromosomes. And these chromosomes are really important for cell reproduction. So that's the nucleus. The mitochondria, what they do is they're kind of like the energy creators of the cell, the power plants of the cell. So what they do is they, take in sugar molecules or fuel, and they pretty much burn it, which creates energy. So this mitochondria creates energy for the cell. Now, the reason all cells need energy is because all chemical reactions in your cell use up some energy. So that's why the mitochondria needs to constantly be at work creating energy for the cells to use. So we've talked about lipids and we've talked about DNA. And we've talked about carbohydrates, because carbohydrates are actually what the mitochondria uses to create energy. So the final macromolecule, if you remember, is proteins. So cells are pretty much like a large part of cell function is to create proteins. So there's a few steps in this process. So pretty much the first step takes place in the ribosomes. And the ribosomes actually take the DNA stored in the nucleus and kind of convert it into protein. That's what the ribosomes, and that's their function. So then it moves on to the endoplasmic reticulum. The endoplasmic reticulum is kind of like a sheath around the nucleus. It's actually stuck on the wall of the nucleus, and it creates new proteins. You can think of the endoplasmic reticulum as like a transport system. It actually, like, because it expands so far, or so it has such a large volume inside the cell, it can transport things really easily. So after it goes into the ribosomes, the protein gets created, it goes into the endoplasmic reticulum, and the endoplasmic reticulum kind of transports the cell, or transports the protein all over the cell. Then the proteins are stored in Golgi bodies. And these Golgi bodies are part of a larger organelle called the Golgi Apparatus. So this Golgi Apparatus contains these things called vesicles. And these vesicles you can think of as tiny cells. They're like really, really small. They have a plasma membrane, they have cytoplasm, and they store these proteins. That's their only function. So they're just like tiny little membranes that store proteins. So that's how a protein is kind of made. It goes from the ribosome to the endoplasmic reticulum to the Golgi apparatus, and then it stays in the vesicles. So another organelle is the vacuole. So the vacuole is kind of like the waste bag. So it just stores stuff within the cell. It can store starch, water, and it also contains waste a lot of the time. Vacuoles are pretty large in plant cells and very, very small in animal cells. That's really important. And also the final organelle is lysosomes. So what lysosomes do is they contain enzymes to break down food particles, waste, and worn out organelles. So you can think of the cell like a machine, right? It has this really smooth function that it needs to keep going. And anytime something is out of place, that pretty much creates a disease, right? So like cancer, the cell reproduction is going really, really fast because the cell is lacking some proteins. And what this does is it actually causes the cell to reproduce really, really fast, which is what causes cancer. So anything that doesn't work becomes a disease. So that's why lysosomes have to digest worn out organelles, because worn out organelles can sometimes go crazy and cause diseases. So that's why lysosomes are really, really important. So plant cells and animal cells are different. So if you remember, we said that plant cells have what we call a cell wall. And this is just a cell wall that surrounds the plasma membrane of the cell. It's a really rigid structure. It's there for support. And it is made up of cellulose, which is, if you remember from our macromolecule discussion, it's a polymer of glucose that's used in plant cells for structure. So another really important thing about plant cells is they also contain something called chloroplasts. And chloroplasts, if you know what photosynthesis is, they carry out photosynthesis. So what photosynthesis is pretty much it absorbs energy from the sun and converts it into sugars. And that's how plants pretty much produce their food. So the really important thing about plant cells is that they have that cell wall made of cellulose. They have the chloroplast, which carries out photosynthesis. And they also have giant vacuoles. That's really important. So we can do this activity here. So it's just pretty much just match parts of the cell. So I don't know if you guys just want to write it out, or just for the first one, where instructions for the cell functions originate. Just type your answers in the chat, just the letter of which it corresponds to. Yeah, okay, so we don't have much time, so I'm just going to go quickly, but yes, it's C, it's the nucleus. Remember, the nucleus is the brain of the cell. So the next one is, they didn't really talk about, so I'll just explain it, where lipids such as steroids are assembled. So you know how I said in the endoplasmic reticulum here, this creates proteins, but there's actually two divisions of the endoplasmic reticulum, what we call the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the rough endoplasmic reticulum contains the ribosomes, and that's the one that kind of, creates proteins. The smooth endoplasmic reticulum actually makes the lipids. So for this answer, it would be smooth endoplasmic reticulum. I didn't explain that, but yeah, smooth endoplasmic reticulum makes lipids. So stores starches and water. All right, so to those of you who got it, good job. It was G, or vacuole. And remember, the vacuoles are giant in plant cells, not so large in animal cells, and they store like waste and starches and water. So assembles proteins. All right, so the answer here was F. If you remember that DNA gets converted to proteins in the ribosomes. Okay, so the region between the cell membrane and the nucleus, which one is that? Yeah, so the answer here was A. We didn't really talk about the nuclear membrane, but you can remember that just a good rule of thumb is that all organelles have membranes. So the nuclear membrane is kind of like the cell membrane, but for the nucleus. So the nuclear membrane would cover the cell pretty much. So next one, transform sugars into usable energy. Yep, so the answer here was D, the mitochondria. You can think of the mitochondria as the powerhouse of the cell. That's a terminology that a lot of people like to use. And remember, mitochondria create energy from sugars. So next one, organelle that stores proteins. Yep, so process of elimination, the answer was B, the Golgi body. Remember, they just store proteins in the vesicles. So now let's look at a diagram. I don't think we actually talked about what each looks like, so I'll just go through it. So this is an example of a plant cell, and the way you can tell is because of the giant vacuole in the middle here. So for that reason, it's a plant cell. Animal cells would not have that. Then remember, you can look for other organelles that plant cells have. For example, this is a chloroplast. You can tell because it's green. You can see how it's kind of like striated with all these lines. That also suggests that it contains chlorophyll. We'll talk about that more when we talk about photosynthesis, but just remember, anything green like that is probably going to be a chloroplast. And then remember, you have the cell wall, which is the thick green thing here. And then you have the kind of plasma membrane, which is much thinner. So that's how you can tell it's a plant cell. So the important things about the plant cell, remember the thin line here is the plasma membrane. The thick green line is the cell wall. The large space like this is the vacuole. This is the chloroplast. This green fluid that everything seems to just be sitting in is the cytoplasm. And then this is the mitochondria. The mitochondria will always look like this because they have these infoldings. I don't know if you can see that really clearly, but they kind of have all these little spikes on them. That's how you know it's a mitochondria. Then this is the nucleus. You can tell because of the DNA wrapped up like this. And then I don't think they really talked about it, but outside here is this is the endoplasmic reticulum. You can't really tell if it's smooth or rough because, I mean, it all looks like smooth because there's no texture on it. If it had texture, it'd be rough. Then in this area right here, this orange thing, this is the Golgi Apparatus. I mean, that's kind of hard to tell, but pretty much the Golgi Apparatus is like, it's very similar looking to the endoplasmic reticulum. It's just a bunch of lines kind of curving in and out. But you can differentiate them because the endoplasmic reticulum is always connected to the nucleus. So the next question is the cell you labeled a plant cell or animal cell. It's definitely a plant cell because of the fact that it has the vacuole, cell wall, and the chloroplasts. Okay, so those are all the cell organelles. If you are a little bit unsure of what we just said and you're kind of confused, don't hesitate to ask. It's a lot of information. Just type your questions in the chat and I will answer. Oh, what is the purple thing? So it's kind of like, so in DNA, when we have DNA, there's kind of like different regions in the nucleus. This is a little bit advanced, but like you see this thick, really blue thing here. That's what we call the nucleolus. It's kind of like a little part of the nucleus. And what it actually does is it gets the DNA ready to be turned into protein by the ribosomes. Because remember, ribosomes create protein. So this purple thing is probably just the nucleolus or some kind of region in the nucleus. That's what I would say. I hope that answers your question. Okay, so let's talk about bacteria and viruses. If you remember that bacteria are unicellular organisms because they're just smaller, and animals and plants are multicellular organisms. And pretty much what's really important about multicellular organisms is they have specialization in their cells. Like we have skin cells, we have muscle cells, we have bone cells, all these different kinds of cells. But like bacteria, which have a single cell, are not going to be very specialized because they just kind of need to live rather than, have specialized function for each of their cells. And cells, these bacteria cells are very, very, very small. Even compared to our cells in our body, bacteria cells are just generally smaller. So because they're just generally smaller, we actually call them, there are different branch of cells called prokaryotic cells. And prokaryotic cells, okay, they don't talk about the other. So prokaryotic cells are like the earliest life form of cells. So when cells first formed, they were prokaryotic, but then they started to evolve and they became what we call eukaryotic. So eukaryotic cells are what make us up, and prokaryotic cells are, kind of what make bacteria and archaea and other, smaller organisms up. So prokaryotic cells versus eukaryotic cells, the biggest difference between prokaryotic cells and eukaryotic cells is the fact that eukaryotic cells have a nucleus and prokaryotic cells do not. And this actually comes from the name. This is how we define them, because eukaryotic, karyo, K-A-R-Y-O, which makes up eukaryotic, actually means it has a nucleus. And then prokaryotic, pro means before, and then karyo means nucleus. So it was before the bacteria actually evolved a nucleus. So that's what differentiates between prokaryotic and eukaryotic. So bacteria, some bacteria can be really bad for you because cholera or other diseases can be caused by bacteria, but many are actually really helpful to us. For example, milk being turned into yogurt is with bacteria. Bread rising is also with bacteria, and they can actually be used for a variety of medicines, and they can produce oxygen in the world. Harmful bacteria can cause a lot of diseases. So bacteria have a bunch of characteristics, one of which is that they don't have a nucleus, nuclear membrane, or any other organelles. So if you remember, we talked about all of those organelles that we had. Bacteria don't have any of them. Pretty much, if you look here, you can see that all they have is a cell membrane or a cell wall outside. Then they have a cell membrane. They have their cytoplasm, and they have chromosomes. That's pretty much all they have. Bacteria actually also have ribosomes. It's not shown here, but they do have ribosomes. So that's prokaryotic cells are much, much simpler. Eukaryotic cells have like layer upon layer of specialization with the nucleus, mitochondria, and all of those other organelles. So another characteristic is they have a single large DNA molecule. And so we call that a circular chromosome. So in our DNA is kind of arranged in a line. So it's just like a really long line with two ends. Bacteria have DNA in a circle. So it's just like a piece of DNA in a circle. And that's what makes up their DNA. And they're really, really, really small. They're much smaller than human cells because they're prokaryotic. So bacteria, they have something called flagellum. So what flagellum are is kind of like a tail on the end of the bacteria that allows it to move. So if you think of like a fish swimming, you can think of like the tail flipping back and forth. Similarly with the flagellum, it makes that like waving motion, and this allows them to move in wind or water. It's not very powerful, so a lot of the times they just go wherever they're carried, but you know they do have some movement. Yeah. Another thing is that bacteria, they also, or not bacteria, but some other cells, human cells, eukaryotic cells, have something called cilia. And what cilia are, you know how we said like flagellum are like a tail? Cilia are like a bunch of little hairs on the end of the bacteria that also allow it to move. But you can think of like cilia as just like small hairs that kind of just come off of the bacteria or the cell, and they move in like a waving motion back and forth. No, we don't need to do this. Okay, so let's talk a little bit about viruses. So coronavirus, we're going to talk about kind of how it works. So pretty much the virus starts attacking your body when it attaches to a cell, which we call the host cell. So when coronavirus infects your body, it will, the first thing it will do is it will attach to a cell and in your body. And then what it does is it releases its genetic material into that cell. So the way it does this is the virus will bind to your cell's plasma membrane, and then it will break open the plasma membrane and then release its material inside. Then what it does is that genetic material gets turned into protein by the ribosomes. So it gets turned into the protein, and then these proteins form new viruses inside the host cell. Then once there are enough viruses in the host cell bursts, and then it releases all the new viruses. So that's pretty much how all viruses work. Just an important thing to note, the virus itself does not or cannot do this process on its own. That's why it infects us, and it uses us as like a host. So it'll use our cell's machinery, its ribosomes, the nucleus, all of that stuff. It'll use it to create new copies of itself. So pretty much the virus attaches to our cell and just hijacks its machinery. That's how viruses work. Okay. Yeah, we don't have to do this. We can do that later. Okay, so the next thing that we're going to talk about is diffusion and osmosis. So diffusion and osmosis are really, really important. It's the way that materials can move in and out of your cell. So remember that cell membrane, the plants have like a thick wall around their cell called the cell wall. And then both plants and animal cells have that cell membrane. So the membrane, what it does, remember I said it was selectively permeable. It allows certain things in and only certain things out. So it controls the flow of molecules in and out of the cell. So the cell membrane kind of has like these small pores across its surface. So it's like these little holes in like a fence. And so the smaller the molecule, the more easily it will be able to pass through these holes. So the easier it can move in. But materials that are very, very large, they cannot move in. And so because we have some molecules moving in and some not being able to move in, we call it selectively permeable. So the cells in our bodies are all selectively permeable. And so we call the or the movement of molecules across the membrane, it's called diffusion. So what is diffusion and how does it work? So pretty much like if you've ever, like if you've been in a kitchen and, someone's cooking something and, that smell, it starts to fill the entire room. So that aroma kind of like starts at, the pot and then it spreads throughout like the rest of the house pretty much. So the closer you are to the pot, actually, the stronger the smell will be. I mean, that just makes sense, right? Because like if you're very, very close to the pot, you're gonna just smell more of the gases coming off. So molecules, just in general, tend to move from areas of high concentrations to low concentrations. And this is the general principle of all of diffusion. And what this means is that, going with our pot example, molecules near the pot are going to be in a very high concentration, right? That's why when you're really close to the pot, you're going to smell a lot of that meal. So, but when you move farther and farther away, you're not going to smell it as much. And so this is because the molecules originate from the pot and then they start to spread throughout the room. And again, this is that same principle, moving from a high concentration to a low concentration. So, they kind of just spread, molecules just generally just spread and occupy available space. And pretty much what they try to do is they try to obtain something called equilibrium. So equilibrium is when the concentration of molecules anywhere is even. So for example, with the pot example, the molecules, they want to spread such that, if you're 5 feet away from the pot, you're still going to smell the same amount if you were right next to the pot. That's why molecules spread. Does everybody understand how things move in diffusion? If you have any questions, just ask. Okay, so let's move on. So this is kind of like an example of diffusion here. So you have these dye particles, and pretty much you have a membrane that's permeable to the water and the dye. So what's going to happen is first these dye particles are really, really concentrated on one side of the membrane. But over time, the dye particles are actually going to diffuse across the membrane. And then it's going to reach an equilibrium point. So the dye particles kind of go from really concentrated on one side to equal on both sides. So pretty much what happens is the dye particles, they reach a point called equilibrium. And this point of equilibrium is actually when both molecules on both sides of the membrane are completely equal. So, if you have ever, watered a plant and then seen it come back to life, this is an example of osmosis. So osmosis is just the movement of water. So if you remember, we said that diffusion was just the movement of molecules across the membrane. Well, osmosis is actually the movement of water just across the membrane. So for example, if you water a plant and the water, just diffuses into the plant cells. What actually happens is the vacuoles and the cytoplasm take up the water, and they start to swell, and they'll return to their normal shape and structure. So that's what osmosis is. So yeah. So let's talk a little bit about how things are going to move if you place something in salty water or in just plain water. So let's say you have a cell, just a normal cell, and you place it in a solution. Or actually, let's do these questions first. Well, I think they go into that later. So pretty much, let's see here. Diffusion moves large molecules across the cell membrane. True or false? Just type your answers in the chat. Okay, so the answer here, I believe, is false. And this is because diffusion, remember, the cell membrane is selectively permeable. So the larger the molecule, the more likely it's actually not going to move. So that's why we say that diffusion is the movement of small molecules across the cell membrane. So molecules move from an area of high concentration to a low concentration. True or false? Okay, so the answer here was true. And molecules will move from an area of higher concentration to an area of lower concentration. That's just what diffusion is. So 3, no energy is required for osmosis and diffusion. Okay, so the answer here was true. Remember that osmosis and diffusion are passive processes, which means that it requires no energy for the movement of molecules. Next, if the salt concentration inside a cell is 2%, the salt concentration outside the cell is 6%, water will flow into the cell by osmosis. If you guys have any questions, just type them in the chat. Okay, so the answer here was, wait, I don't remember what the answer was. Okay, so the answer was false, actually. And so this is what we're going to get into. But pretty much, salt concentration inside a cell is 2%, and the salt concentration outside the cell is 6%. So what's going to happen is water is actually going to move to the outside of the cell. And the reason for this is based on the concentrations, right? So, if it was asking where is the salt going to move, then the salt would move inside into the cell because, the salt concentration is higher on the outside than it is on the inside. But you have to remember that salt is not the only thing remember how we said there's a cytoplasm, and so that actually that's just water, and that's what's contained in the salt. So what's going to happen actually is the water that's inside the cell is going to have a, It's going to have a low concentration of salt. And the salt concentration outside of the cell is going to be very, highly concentrated. So what this means is that the salt concentration, the water concentration inside the cell is going to be very high, and the water concentration outside the cell is going to be low. And the way I came to that conclusion is because the only two things that make up the solution, based on what we're given, is salt and water. So if salt concentration inside is 2%, that means water must be the other 98%. And if salt concentration outside is 6%, that means water must make up the other 94%. So now we have inside the cell, the water concentration is 98%, and outside the cell, the water concentration is 94%. So that means that the water is actually going to flow out of the cell by osmosis. So that is how you would figure out that problem. So I'm just going to go onto a new slide set here. So we're going to go into a little more detail here about diffusion and how we can define it, and we'll also look at that problem that I just explained, and we'll see another way we can define it. So just a quick review, if you remember from one of our earlier discussions, a solute is the thing that gets dissolved. So in that question 4, the example was salt. The salt is getting dissolved in, and then it gets dissolved in the solvent. And the solvent is the water. Remember, water, one property of water is that is that it is a universal solvent, which means it can have a lot of things dissolved in it. The other aspect is the solution. So the solution is kind of like the solute dissolving in the solvent. An example of this is ocean water. Ocean water contains the salt, and the salt is the solute, water is the solvent. And so the concentration. So how do we figure out the concentration? So the concentration is the percent represented by either the solute or the solvent. So in that previous example, we had the solute concentration being 2% on one side, 6% on the other, and the solvent concentration was 98% and 94%. So that's the concentration. Remember that concentration is kind of just like the amount of salt over the total amount of substance. So in that case, when they said there was 2% salt, that means that the amount of salt over the total amount of salt and water was 2%. That's how they came up with the concentration. So this is what we learned about diffusion. Remember, if you have a solute here, it will, kind of spread out. So this is similar to what we said with the coating. So assume that the pot is over here. The pot is going to release a lot of these air molecules or solute, and pretty much what's going to happen is that's going to diffuse throughout the air, which is the solvent. So this, each of these red is like an aroma, I guess you could say, and that's kind of diffusing throughout the air. So we talked about this. So now let's talk about cells. So we have to define 3 terms. One of them is hypotonic. And what hypotonic means is pretty much a situation where the solution has a lower concentration of solute outside the cell compared to the concentration of solute inside the cell. So for example, salt, let's take our same example with salt. So if you had 2% salt outside the cell and then 2% salt inside the cell, 6% salt inside the cell, that means you would have a higher concentration of salt inside the cell, and you would have a lesser concentration of salt outside the cell, which means you would have a hypotonic solution. So, pretty much the example that they're showing here, you can see that this is the cell. It has a really high concentration of the solute, and you can see it because the red dots are really, really concentrated here. Then you have this water outside, which is brackish, And you can see that the red dots are not as concentrated, meaning just the concentration of the red dots is less. So you have higher concentration here, lower concentration here. So the cell is in a hypotonic solution. So when a cell is in a hypotonic solution, what actually happens is the water is going to move from outside the cell to inside the cell. So why? So what actually why does this occur? And this is because water is in a higher concentration out here and a lower concentration over here. And so we know that that's actually the opposite of salt. Salt was higher concentration in the cell, lower concentration out of the cell. But water is actually in a higher concentration out of the cell and a lower concentration inside the cell. So what's going to happen is water is actually going to move into the cell to balance out this concentration gradient. So what could happen is if the concentration gradient is steep enough, so much water could move into the cell that your cell could burst or explode because it has so much water. So the other topic is hypertonic. And a hypertonic solution is the opposite of hypotonic. So remember, in hypotonic, you had high concentration of solute inside the cell and low concentration outside. In a hypertonic solution, you have a high concentration of solute outside the cell and a low concentration inside. So what's actually going to happen is the water is going to move from inside the cell to outside the cell. And so again, why does this occur? We can think of it in terms of water concentration. So outside the cell, you're going to have a very, very low concentration of water. But inside the cell, you're going to have a higher concentration of water. So what's going to happen is the water is going to move out of the cell. And pretty much because there's nothing kind of like holding the cell open, it's just going to collapse because like there's not enough water. So as you can see in the diagram here, there's kind of no concentration of solute here. There's a very high concentration outside. So it's a hypertonic solution and water is going to move outside. A good rule of thumb to remember is that water always moves from a hypotonic solution to a hypertonic solution, which means that water will move where there's a low concentration of salt to where there is a high concentration of salt. That's just generally how water moves. So now let's talk about isotonic solutions. So isotonic solutions is when the solution actually has an equal concentration of solute inside and outside. So as you can see here, all of the dots are kind of evenly spaced. Concentration is the same inside and out. So pretty much nothing happens. The no water, or there will be no net movement of water. And so the water will kind of just be moving back and forth, but nothing special is going to happen. Okay, so now let's talk about something called facilitated diffusion. So remember all of this, isotonic solutions, hypertonic, hypotonic, they all involve the movement of water, which was osmosis, but they all didn't require energy. So now let's talk, well, actually, these still don't require energy, but facilitated diffusion is when the molecules actually move through carrier proteins. So you can see here, this is your plasma membrane. So these are all the phospholipids. The phospholipids are arranged like this in this bilayer. So what actually happens is you have these carrier proteins kind of in the middle of the bilayer, and they actually facilitate, which is why it's called facilitated diffusion. They facilitate the movement of molecules across their concentration gradient. An important thing to remember, though, is facilitated diffusion does not require energy. It's still moving the molecules from high to low concentration. Anything that requires energy is moving things against its concentration gradient. So the concentration gradient always goes from high to low. So anything that's moving from high to low concentration does not require energy. anything that's moving from low to high concentration requires energy. So examples of this, organisms moving glucose into in the bloodstream. This is facilitated diffusion. The glucose actually just diffuses into the red blood cell through a carrier protein in the plasma membrane. Now, active transport. So active transport requires energy. So active transport actually moves molecules from an area of lower to an area of higher concentration. And remember, this is the opposite of what we want. We want it to go from an area of higher to lower concentrations. That's what's natural. So because it's not natural, you have to use energy to do so. So therefore, you have to use proteins and energy. So if you remember here, we just use proteins in facilitated diffusion, and then all of this osmosis and just normal passive diffusion required no proteins to diffuse to the cell membrane. But with active transport, you need energy and you need a carrier protein. So carrier proteins and these energy molecules kind of pump these molecules from a low to high concentration. So interestingly, in fish, the salt concentration in the ocean is very, very high. So what you're actually going to see is you're going to see water trying to almost escape from the fish because the concentration outside is so high. So actually, and also the fish are going to be taking up a lot of salt because when they drink the salt water, they're going to have a lot of salt in their system. So they actually have an adaptation for this, and it uses active transport. And what actually happens is they pump the salt out of the body and back into the water using active transport. So let's just quickly go back to this quiz, or the one that we did before. I just want to make sure you guys understand. So I just want to see you guys just do question 4 and tell me which cell is, or which area is hypotonic and which area is hypertonic. Okay, so for this problem, what we actually see is that the solution inside the cell is going to be hypotonic and the solution outside the cell is hypertonic. So here the answer was false, if you remember. So what's going to happen is the water, remember we said it always moves from hypotonic to hypertonic. So the inside of the cell is hypotonic, the outside of the cell is hypertonic. So the water is going to move from inside to outside the cell, so the answer here would be false. So I have a few more questions. So here, question 1, which of the following is not a type of passive transport? So the answer here was endocytosis. So remember we said that diffusion and osmosis require no energy and no proteins. Facilitated diffusion does not require energy but uses proteins. Endocytosis we haven't talked about yet, but endocytosis is pretty much when a cell kind of engulfs material from the outside. So an example of this is with your immune cells. So what actually happens is your immune cells kind of, if you will, eat bacteria or it comes in contact with. So it uses a process called endocytosis where it eats things. So this requires a lot of energy. And passive transport is when you require no energy, and active transport requires energy. All right, so question 2. Chamber A contains 40% helium and chamber B contains 20% helium. Chambers are connected by a tube. The molecules are free to cross. Which of the following will occur? Okay, so yeah, the answer here was A, or the first choice. So the helium is going to move from chamber A to chamber B. Remember, molecules always move from a higher concentration to a lower concentration. In chamber A, you have a concentration of 40%. Chamber B, it's only 20%. So the molecules are going to move from chamber A to chamber B. Okay, what will happen to an animal cell placed in a saltwater solution? Okay, so the answer here is A. And remember, if it's placed in a saltwater solution, that suggests that the solution is hypertonic. And remember, water will always move from a concentration, or it will move from a hypotonic to a hypertonic solution. So the cell in this case is hypotonic, the outside is hypertonic. So water is going to move from inside to out, which means the cell is going to shrink because it has less water. Okay, next question. An animal cell placed in a hypotonic solution will. So if an animal cell is placed in a hypotonic solution, it's going to gain water. Same principle as before. If it's placed in a hypotonic solution, that means it's hypertonic to the outside solution, which means water is going to move from hypotonic to hypertonic or outside to in, which means it's going to take on water. Okay, so number 5, which of the following is a type of active transport? Sodium potassium pump, endocytosis, exocytosis, or all of these? Okay, so the answer here was D. If you remember, pump is the key word for sodium potassium. Remember, anything that pumps a molecule suggests that it's using active transport. And remember, I said endocytosis is the process by which molecules are engulfed that requires energy. Exocytosis, I actually have a diagram here to show you guys what these are. So this is kind of all of the different types of movements. So we talked about endocytosis, which is the process of taking material into the cell. There's also exocytosis. and this is the removal of materials from a cell. So if you remember when we talked about the cell, we said that the proteins are stored in something called vesicles. So one way vesicles can actually be used is they can actually be kind of taken out of the cell by exocytosis. And so pretty much the vesicle will bind to the plasma membrane and kind of excrete its contents. And so the reason it will do this, for example, if you guys know about insulin, insulin is like a really major molecule to absorb blood sugar from the bloodstream. It's created by the pancreas. So the pancreas cells, they create the insulin. Then what happens is they're stored in the vesicles. When they're needed, they're released from the cells by exocytosis into the bloodstream. So that's how proteins make, or how cells make proteins and then release them. The other type is phagocytosis. And remember how I said that the immune cells can take a bacteria through endocytosis? Well, if the bacteria cells are especially large, they're going to use phagocytosis. And phagocytosis is kind of like, or phago, P-H-A-G-O, in Greek means to eat. So they're literally eating cells. So it's just a type of endocytosis, but they're pretty much taking up much, much larger molecules. Then another type is penocytosis, and penocytosis is cell drinking. Peno means drink in Greek, so penocytosis means cell drinking. So the cells will just incorporate and engulf little drops of fluid into the cell. So that's how penocytosis works. Okay, so back to the questions here. So yeah, the answer to this was all of these. Penocytosis, phagocytosis, exocytosis, and endocytosis all require energy. And then remember, the sodium potassium pump also requires energy. So active transport requires, it should be pretty easy now. So the answer here was C, or energy. Remember, passive transport, for the first question, passive transport does not require energy. Active transport is any type of movement that requires energy. Okay, cool. Okay, so those are the big topics for cell biology, diffusion, and osmosis. We're going to move on now to cell respiration. If you are still confused about cells or anything like that, don't hesitate to ask, because we're just kind of building on the knowledge we already have. So please ask. Review the lecture when it's over on the YouTube channel. If you have any questions, you can always email me afterwards too. So cell respiration, right? So if you remember, we said that mitochondria create energy from sugars. So now we're going to talk about the specific process by which they do this. So overview of respiration. So what is cell respiration? So cell respiration is the process by which organisms use energy from food. And so they're going to use glucose, which is a sugar. They can also use fats sometimes, and they fuel the synthesis of ATP. So now this is the equation for cell respiration. Glucose, which is the sugar with the formula C6H12O6, plus 6 molecules of oxygen gas will create 6 carbon dioxide, 6 water molecules, and ATPs. So ATPs, we like to think of as the energy currency of the cell. So think of like the more ATPs a cell has, the more energy it has. ATP is just a molecule and it stores energy. So this is the equation. Glucose plus 6 molecules of oxygen gas creates 6 molecules of carbon dioxide plus 6 molecules of water and energy. So important thing is that Cell respiration requires oxygen. Also, another really important thing is that plant cells also use or also have cell respiration. If you remember that we said the plant cells had chloroplasts, in which they took the sun's energy and they built back sugars. Well, these same sugars are being transported to mitochondria to create ATP. So what this does is it provides a supply of usable energy for the cells. So the glucose molecule itself, cells can't really use that for energy, but ATP they can use. So that's why they turn it into ATP. Okay. So cell respiration in the human body. So the cellular respiration actually takes place in the mitochondria. But there's another type of respiration, which means breathing. Respiration just means breathing. Cellular respiration is the process by which glucose is turned into ATP. Respiration is the process by which we breathe, pretty much. So these are like the important steps, right? The oxygen is going to come from our lungs and it's going to go into our cells. We're going to use that for cell respiration. When we're done, our cells are going to release CO2 back into the lungs, which are going to be, or it's going to be excreted later. And the sugars that we get come from digestion, from our eating, going to be turned into glucose, which is usable for our cell to create energy. So there's three main steps of respiration. Don't be intimidated by this diagram. We're going to break it down. So glycolysis is the initial breakdown of glucose. So again, let's look at some roots here, our Greek roots. So glyco, G-L-Y-C-O, means sugar. Like glycogen, if you remember, was the sugar that stores energy in animal cells. So that's glycogen. And then lysis, L-Y-S-I-S, generally means to break. So anything with lysis just means to break. So this first step means breaking sugars, literally breaking sugars. The next step is the citric acid cycle. It can also be called the Krebs cycle, and it can also be called the tricarboxylic acid cycle. So it has a ton of names, and it pretty much completes the breakdown of glucose. And the final step is oxidative phosphorylation. And this is where electrons are transported through the electron transport chain. And this generates a proton gradient, which produces ATP, and we'll get into how that works later. So I want to talk about this molecule, or NADH and FADH2. So these are energy carrying molecules. And so what they do is they carry these high energy electrons from the other two steps to the electron transport chain. So the glycolysis will produce these NADH molecules. The citric acid cycle will produce the NADH and FADH2 molecules, and they will all be moved to the electron transport chain. So let's talk about glycolysis. So glycolysis is a series of chemical reactions that breaks down glucose into 2 3-carbon molecules called pyruvate. So if you remember from here, or from the equation, glucose is a 6-carbon molecule. It has 6 carbons. So the first step of cell respiration, glycolysis, is to split the glucose molecule into 2 3-carbon pyruvate molecules. This is the first step. It occurs in the cytoplasm of the cell. So this first step does not occur in the mitochondria. That's important. The first step of cell respiration occurs in the cytoplasm. Glycolysis does not require oxygen. It doesn't need any oxygen. So another important thing. So let's talk about what it takes up and what it makes. So glycolysis, starts with a single glucose molecule, and it will create 2 ATP, which remember is the energy currency. So after glycolysis, we already have 2 ATP molecules, and then it'll also, make that energy carrying molecule called NADH. So if you look here at this step, you can see that two NAD plus are being converted to NADH. So what this means pretty much is that electrons are pretty much being taken up by this NAD plus molecule, and they become an NADH molecule. And that's how the NADH will carry electrons. So whenever you see NADH, that means that it's in the form where it's carrying electrons. NAD plus is in the form where it's ready to take up electrons to be carried. So after NADH will deliver its electrons to wherever it needs to go, it reverts back to NAD plus and now is ready to take up electrons again. NAD plus, after it takes up electrons, can be converted back to NADH. Now, another important thing here is you'll see that ATP is converted from ADP into ATP. So the reason that the first molecule is ADP and then gets converted to ATP is because ATP contains phosphate groups, which is using the molecule phosphorus. So ADP stands for adenosine or diphosphate. ATP stands for adenosine triphosphate. So think about the name adenosine di, which means two phosphates, and then adenosine triphosphate, which means it has three phosphates. So pretty much what's going to happen is the ADP molecule is going to take up a phosphate and become ATP. So that's how ADP gets converted to ATP. So the final products of glycolysis, 2 ATP, 2 NADH, and these two really important pyruvate molecules. So let's talk about fermentation. So some of you might know fermentation through yogurt. Yogurt is formed from fermentation. Another really important part of fermentation is creating bread. Bread is created through fermentation. So when there's not enough oxygen, what happens is we go into fermentation. So in your muscles, like after you sprint or are going for a long distance run, you'll feel like your legs hurt and you'll get like that burning sensation. And that is due to the products of fermentation. So, when you're running, your muscles are starved of oxygen, but they still need the energy, that ATP. They start to go into fermentation. So pretty much what happens is there's two types. There's lactic acid fermentation and ethanol fermentation. Lactic acid fermentation is what we use when you're running. So the muscles are starved of oxygen when you run. So if you've ever ran, I'm guessing probably all of you have, you've known that feeling of being really, really tired. And the reason that you're tired is because your heart is pumping really, really fast. Remember, the heart is part of the circulatory system. It's moving the blood around your body. The blood carries oxygen. So when you're really tired, that suggests that your heart kind of is not pumping the blood fast enough to your muscles to meet the oxygen demand. So in that sense, the muscles do not have enough oxygen to carry out the cell respiration, so they're forced to revert to citric acid cycle. So it's all based on your heart. So because your heart doesn't pump enough blood, muscles don't have enough oxygen, so they have to go into the fermentation. So lactic acid fermentation is in animals. Ethanol fermentation is in yeast. So yeast actually is put in bread. So when you eat bread, these yeast actually use fermentation to create CO2, which actually makes the bread rise. So, but the thing is, it results in a relatively low energy yield. So remember I said, right here, each step of glycolysis only yields 2 ATP. So that means that fermentation is kind of like an extension of glycolysis, because there's no oxygen, and glycolysis, remember, does not require oxygen. These cells start to kind of use glycolysis as their primary source of energy. They don't go into the any of the other steps. So what actually results is a low energy yield. They just get the energy from glycolysis. And if you remember, glycolysis produces 2 ATP, so they're only going to get 2 ATP per glucose molecule that they start with. So, but the important thing about fermentation is that it has this important step, and that's, they define fermentation as that process after glycolysis. Some people define it as with glycolysis, but pretty much pyruvate that, remember I said pyruvate was formed from glycolysis, it gets turned into another product, and that is either lactate here or ethanol. So yeast will turn it into ethanol, we will turn it into lactate. But the important thing is that NADH gets recycled to NAD+. So remember in glycolysis, NAD+ gained its electrons, got converted to NADH. In fermentation, NADH gets reverted back to NAD+. And this is really, really important because if you just did glycolysis, you would have a ton of NADH buildup. You have no more NAD+. But with fermentation, you're going to recycle that NADH back to NAD+. Now, if you remember from this diagram here, we said that the NADH produced by glycolysis goes to the electron transport chain. So that means, but the electron transport chain actually requires oxygen. So that's why the NADH has to be recycled, because the only way it could be used up is in the electron transport chain. That's not happening because we have no oxygen. So it's forced to go into fermentation to recycle this NADH. So that's what fermentation is. So let's now talk about the citric acid cycle. So the citric acid cycle occurs in the matrix. So the matrix, you can think of as the inner space of the mitochondria. So look at the structure of the mitochondria here. It has two membranes. The outer membrane, you can see has the yellow coating here, and the inner membrane. Remember how I said that on a diagram you can always identify the mitochondria because it has like these foldings like this. And so when you actually look at like inside a mitochondria, it actually looks like that. You have these infoldings of the inner membrane. The outer membrane is smooth. So inside the inner membrane, it contains a compartment called the matrix. The matrix is where the Krebs cycle takes place. So the Krebs cycle, or the citric acid cycle, whatever you want to call it, takes these 2 pyruvate and kind of recycles them. So I don't know if they have a diagram. Okay, they're here. So the first step of the citric acid cycle is this pyruvate that we have is converted into a molecule called acetyl-CoA. So acetyl-CoA is made up of an acetyl group and a CoA molecule. So you don't have to know this in this much detail, but it just makes more sense this way. So pyruvate is converted into acetyl-CoA. Then CoA is kind of like an energy molecule. So because once it breaks off of the acetyl group, it actually gives it a little bit of energy to do the entire cycle. So it starts with this 2-carbon molecule. Then what happens is, 3-carbon molecule gets converted into acetyl-CoA, CO2, and then it goes through these steps. The important thing about the citric acid cycle is this is where the CO2 is going to be produced. You're going to have 2 CO2 produced here. Then in the conversion to acetyl-CoA, you're going to have another CO2 produced there. You're also going to have 3 NADH be converted, or 3 NAD plus be converted to NADH and given those electrons. You're going to have 1 ADP being converted into ATP. Then you're going to have 1 FAD being converted to FADH2. Now, FADH2 is like the same thing as NAD plus. FAD is the same thing as NAD plus. NADH, same thing as FADH2. They both have the same purpose. They transport electrons. It's just that their structure is a little bit different, but you can pretty much remember them as the same thing. They serve a very, very similar function. So the important thing about this citric acid cycle is that you have to look at, like when you're doing problems or anything like that, you have to look at how many pyruvate are being entered into the cycle. This is like a lot of teachers will put it like this is a question on test, because they'll ask you or they'll give you the cycle and they'll say how many molecules of pyruvate are being put in. So if you remember from glycolysis produces 2 pyruvate molecules. But this diagram here actually shows the conversion of only 1 pyruvate molecule. If you remember from our equation, here, it says that 6 CO2 should be produced by the breakdown of 1 glucose molecule. But here, it shows us that only 2 CO2 are being produced here and 1 CO2 is being produced here. So this actually means that we're 3 CO2 short because you only have 1 and 2 here. But the thing is, we're actually only using 1 pyruvate in this cycle. We actually have another pyruvate still left over from glycolysis. So if we put that into the cycle too, it's actually going to create another 3 CO2. So the end products are, I don't know why it says 8 here, it should be 6. But it pretty much creates 6 NADH. It'll create 2 FADH2, 2 ATP, and 6 CO2. Be careful, if it's 1 pyruvate, it's only going to create 3 CO2, 3 NADH, 1 FADH2, and 1 ATP. If it's 2 pyruvate, it's going to create 6 CO2, 6 NADH, 2 ATP, and 2 FADH2. So now the final step of respiration is called oxidative phosphorylation. So oxidative phosphorylation is a little more complicated. It contains what we call the electron transport chain, is the first step of oxidative phosphorylation. And so the term oxidative phosphorylation, you can think of oxidative as requiring oxygen. It's like oxygen. So this requires oxygen. And then phosphorylation. So if you remember when I talked about ADP, if you combine that with another phosphate, it forms ATP. So that's what phosphorylation is. It's the addition of a third phosphate that will end up creating ATP. So it occurs in 2 stages. So the first step of oxidative phosphorylation is the electron transport chain. So the electron transport chain kind of, if you remember, we talked about NADH and FADH2 as having those really high energy electrons. What it's going to do is it's going to take those electrons and it's kind of going to transfer them along like a pathway. So you can see, I mean, this is kind of a weird diagram, but you can see the electrons are right here. It's 2E minus. So what they're going to do is they're going to be transported all along these protein complexes like this. So, there's 4 different protein complexes. This is 1, this is 2, this is 3, this is 4. And it's going to be transported along all of these. And then it's finally going to join with oxygen to create water. So that's where the water comes from in the cell respiration equation. It's from the electrons moving. So that's what the electron's function is. So, but let's talk about oxidation and reduction. So this is how the electron is actually moved, because it doesn't just flow through here. It kind of joins onto a molecule, then it unbinds, then it binds onto another one, then unbinds, then it binds onto another one, and that process will kind of repeat until it finally binds onto oxygen. So pretty much oxidation is when you lose an electron or a hydrogen, usually with a hydrogen atom. So when you lose an electron or a hydrogen atom, you can think of that as NADH being converted back to NAD+. That's what we call oxidation. Oxidation is loss, and then reduction is the opposite of that. It's the gain of an electron. So when NAD+ gets converted to NADH, that's the gain of an electron. That's reduction. A good mnemonic to remember this is oil-rig, OILRIG. Oil-rig stands for oxidation is loss, reduction is gain. Oil-rig. And So that's just a mnemonic I use to remember it. So the important part, so how does this relate back to the electron transport chain? Well, pretty much what happens is, remember how I said that, the electron first will get released from the NADH and bind to the first protein. Then it gets released from the first protein, binds to the second, and then so on and so on and so on until it finally binds to oxygen to create water. So this is a reduction oxidation reaction. So what happens is NADH actually becomes oxidized, which means that it's going to lose that electron. But the first protein complex is going to become reduced, meaning it gains the electron. But then this second protein complex pretty much just kind of wants that electron more. So it's going to kind of unbind from the first protein complex, which is oxidation. So the first protein complex is going to be oxidized. It's going to move to the second one. The second one's going to become reduced, and kind of so on and so forth through all of the protein complexes, one being oxidized, one being reduced, and then finally it's going to bind to oxygen, and it's going to create water. So that's why we need oxygen. If you don't have oxygen, this electron is just kind of stuck in the chain, and it kind of backs up the chain because oxygen is not taking it out. So that's why you need oxygen for respiration. And when you don't have oxygen, you can't, do cell respiration because your electron transport chain is just backed up. So another important thing about the electron transport chain is that it works to pump hydrogen ions. So if you remember, we just learned about diffusion, active transport. So the electron transport acts as energy. So as electrons are being transported down this electron transport chain, is actually creating a lot of energy through all of this series of reactions. And because it's creating a lot of this energy, hydrogen is actually being pumped. And it creates what we call a H+ gradient, or a hydrogen gradient. And so that's where the next step of oxidative phosphorylation comes in, and that's chemical osmosis. And remember, osmosis is the movement of water. Chemi is short for chemical. So it's pretty much the movement of chemicals through the same process as osmosis, which was just diffusion. So yeah, we talked about that. So pretty much what's going to happen is you're going to have a very, just to orient you on this diagram here, you're going to have, this is the matrix where the Krebs cycle is taking place. And then this is the, what we call the intermembrane space. So remember we said mitochondria has two membranes. Outer membrane, shown here, this is the cytoplasm out here. This is the intermembrane space. Inter meaning between, so between the membranes, intermembrane space. And you're going to have the inner membrane, which is going to be here. Remember we said over here, the space is the matrix. This is also where the Krebs cycle takes place. So pretty much the hydrogens are going to be pumped from the matrix into the intermembrane space. And this is what's going to create the gradient. So it's going to be pumped from here into here. So now because we're pumping, remember when we say pump, we generally mean from low concentration to high concentration. So because we're pumping from here to here, that means that there's a low concentration of hydrogen here and a high concentration of hydrogen here, which means that the hydrogen wants to move this way. It wants to come back, but it can't because it's being pumped out here. So the energy of movement of the electrons pumps the hydrogen from the matrix into the intermembrane space. So chemiosmosis is the kind of the diffusion of hydrogen back into the matrix. So you can see how there's a really high concentration of hydrogen here, not much hydrogen here. So what's going to happen is the hydrogen from the intermembrane space is going to diffuse back into the matrix. And so this movement from high to low concentration actually creates energy and allows ADP to be phosphorylated to ATP. The molecule that does this is called ATP synthase. ATP being the molecule, and if you know the word synthesis, which means to create, it sounds like that. And then if you remember when we talked about enzymes, we said that the ending ase suggests that this is an enzyme, so that's ATP synthase. So remember, ATP synthase, enzyme that creates ADP plus P to ATP. So the total of oxidative phosphorylation will yield up to 34 ATP per glucose molecule. If you remember from glycolysis, glycolysis only yields 2 ATP. That's why fermentation is really, really inefficient, because it only yields 2 ATP, but oxidative phosphorylation can yield up to 34 ATP, so it's 17 times more efficient. So that's like why we use oxidative phosphorylation. So yeah, this is another diagram. You can see how the NADH transfers its electrons. This yellow path are the electrons. The green is the hydrogen. The hydrogen is being pumped from the matrix to the intramembrane space, and this causes hydrogen to move through the ATP synthase by chemiosmosis and converts ATP into ADP and phosphate group. So just a summary of oxidative phosphorylation. You have the electron carriers, NADH and FADH2 created from glycolysis in the Krebs cycle. This energy is converted into a hydrogen gradient from the electron transport chain. Then the energy stored in the hydrogen gradient via chemiosmosis creates the molecule of ATP. So let's just do this quick activity here. I just want you to just type in the chat, give me like 1-sentence description of glycolysis, and then give me another sentence description of fermentation. Okay, so yeah, glycolysis breaks down glucose, 2-pyruvate, 2-ATP, 2-NADH. That's a good answer. And then if anyone wants to give me a sentence on what fermentation is, I'll gladly take it. So. Right, so fermentation generates ATP without oxygen, generates 2 ATP. So that's a good answer. So the citric acid cycle, anyone wants to give me a sentence for that? Can we get some more participation? I mean, there's 36 of you. So yeah, all you have to do is type a sentence. If you don't understand anything, please ask. I'd be willing to take questions. Okay, so the citric acid cycle, just remember it breaks down the pyruvate molecule, creates NADH and FADH2. So oxidation versus reduction. just give me which is loss of electrons and which is gain of electrons. Yeah, so oxidation is loss, reduction is gain. You can use the mnemonic oil rig to remember that. So oxidative phosphorylation, anyone wants to give me a sentence describing oxidative phosphorylation? Yeah, so oxidative phosphorylation is when we add a third phosphate to ADP. So chemiosmosis. So chemiosmosis you can remember as the hydrogen gradient that moves through the ATP synthase, and then which will create ATP from ADP. The electron transport chain. Anybody have a sentence for that? Okay, so the electron transport chain is pretty much when electrons are transported from the NADH and FADH2, and the final electron acceptor will be oxygen, and this actually creates the energy for the hydrogens to be pumped to one side. And the final one, ATP synthase. I feel like that's pretty self-explanatory. It just creates the ATP using the hydrogen gradient. Okay, so we finished all the material pretty early, and I don't really have anything else. And yeah, so I think we're gonna end it there today. If you guys do have any questions, please ask now before the meeting is over. Just quickly type them in the chat. Okay, so if there's no other questions, then we will end it there. Thank you guys for attending. Remember, next week we're also going to have a long class starting at 6:30. So yeah.